And just very briefly, what accounts for secondary hyperalgesia? Because that's a very important phenomenon that we see in patients or in subjects, and we believe that mechanisms of secondary hyperalgesia are very essential to what is happening in pain patients, because we see similar phenomena in pain patients as well. For example, in pain patients after nerve injury, those patients might actually, besides complaining about spontaneous pain, those patients might actually report that just a light touch similar to sunburn becomes painful. So the neuronal mechanisms of secondary hyperalgesia are very important to explore, to develop better treatment strategies for those patients. And this is a classical experiment done by Bob Lamotte with a capsaicin injection, similar to what I explained previously. So with the injection you get massive pain, but you get also zones of hyperalgesia in the skin. And for one special form of hyperalgesia, the punctate hyperalgesia to punctate stimuli, that hyperalgesia is actually pretty long lasting, over 3 hours. Now in this experiment, what Bob did in his volunteers, what he actually produced a field block. So he anesthetized a peripheral nerve high up on the arm. He got a zone of analgesia at the lower arm, and into that zone he actually injected capsaicin and asked his subject, what do you feel? And he also did sensory testing on those subjects. And first of all, they reported no pain, and secondly, there is no development of secondary hyperalgesia. They still develop a skin reddening, but all the forms of hyperalgesia to light touch or to pinprick are completely prevented. And this basically argues that you need a nociceptive input from nociceptors in the periphery, from the periphery into the central nervous system to produce central sensitization. Again, highlights the importance of what Michael Catarina already mentioned. Trying to silence nociceptors at the periphery is the best, might be one of the best ways to prevent zones of secondary hyperalgesia and to help those patients suffering from it. Another important experiment in that context was performed actually by Eric Thorberg. And Eric is a master, or has been a master in using microneurography in humans, where they basically put a small needle percutaneously through the skin into a nerve. And what you can do with those needles is you can actually record neuronal activity, but you can also stimulate primary afferent nerve fibers. And in this setting, actually, he stimulated such a large myelinated fiber just above the ankle in the superficial peroneal nerve. And what subjects usually report in response to that stimulation is that they feel the sensation of touch, which is consistent with the function of large myelinated afferent nerve fibers. And the next step then, he actually placed the capsaicin injection in such a manner that the receptive field for this primary afferent here on the dorsum of the skin would fall into the secondary zone of hyperalgesia. And then he retested his electrical stimulus, the effect of the electrical stimulus applied at the superficial peroneal nerve above the ankle. And what the subject actually reported, that following the capsaicin injection, there is not only the sensation of touch, but that there is also the sensation of pain, suggesting that now after the capsaicin injection, after this nociceptive input from the skin, that this non-noxious input from large myelinated fibers or touch fibers becomes pain. So the underlying hypothesis, or the model that we have, is basically that in the primary zone, sensitized nociceptors from the skin are able to sensitize central neurons. And that actually leads to ongoing pain as well as hyperalgesia, to mechanical and heat stimuli in the primary zone of hyperalgesia. In the secondary zone, we believe that primary afferents are also able to sensitize central neurons, shown here on the right, that basically innervate the secondary zone in such a manner that these neurons now show an increased input to low threshold mechanoreceptors. Identifying and studying the mechanisms of this central sensitization here on the right is a major goal in pain research, and also identifying non-neuronal elements that could contribute to this sensitization. For example, glial cells, the receptors that actually are expressed on primary afferents or on second-order neurons in the pain pathway. how react to these sensitizing stimuli, what is changing. So this is of central sensitization is of one of the major goals in pain research to better understand the mechanisms. So the summary for what I have shown you today is that there's basically a diverse set of nociceptive afferents and peripheral nerves that respond to different nociceptive stimuli. I've shown you that an increased responsiveness of nociceptive afferents accounts for primary hyperalgesia, and also that central sensitization accounts for secondary hyperalgesia. And that is that an input from non-nociceptive afferents produces pain. Where do I think our field is going? And what is the future of basic pain research, especially in my field where I'm interested in primary afferent nociceptors? And Michael has already pointed this out, that even nociceptive neurons at the DRG level are very diverse. And one of the two biggest populations that have been identified many, many years ago is a population called peptidergic and non-peptidergic. Here on the left-hand side, you see actually the marker for peptidergic neurons expressed in the skin, CGRP. And here on the right-hand side, you see a genetically modified mouse where a green fluorescent protein has been expressed in the non-peptidergic population. And you can actually see that the non-peptidergic population and the peptidergic population seem to differ in their where their receptive endings project. You can see that the non-peptidergic neuron has a much more superficial terminalization than the peptidergic neurons. And there's some evidence from the mouse that these two neuronal nociceptive populations actually might serve two different sensations. And my research in non-human primate, actually, the ongoing research focuses identifying what these two populations do. And what you can actually, when you expose a polymodal nociceptor to a stepped heat stimulus, you can actually identify two different response patterns in polymodal nociceptors. You can see nociceptors which we call quickly responsive neurons. Those are neurons that with the increase in heat start to respond. Some of them show this burst of activity with the onset of the stimulus. And in the second population of what we call slowly responsive C fibers, you can see that this unit actually starts long after the onset of the heat stimulus. We know that the thermal thresholds in those two polymodal nociceptor populations are markedly different. So for the QCs, it's around 40 degrees. For the SCs, for the slowly responsive units, are 45 degrees Celsius. And as a last step, these are very new data. I just want to show you that these two types of populations respond differently to an agonist that we think can for the first time differentiate those peptidergic versus non-peptidergic populations, allowing us for the first time to study the role of these two different populations in human sensation and to see what their response properties are.